Our protocol can be used to improve the quality and quantity of gene expression data generated from FFP-derived RNA samples, especially in suboptimal quality samples.
This technique assesses the RNA quality within FFPE tissue samples and enables optimization of their downstream processing to improve the quantity and quality of the sample sequencing data.
This method allows the comprehensive characterization of a transcript in biological and clinical sample and may provide insight into functional elements of the genome in development and disease.
The FFP-RNA degradation and selection of the methods for sample QC, library preparation, and data analysis can all be very tricky. Be meticulous when selecting the right methods and appropriate controls.
Visual demonstration allows viewers to observe the small, subtle modifications and precautions needed during the planning, assessment, and the sequencing process, especially for poorly preserved FFPE samples.
To check the quality of the RNA sample, run the sample on an RNA QC system according to the manufacturer's instructions, and use the appropriate bioanalysis software to analyze the distribution of the RNA fragments within the sample, and to calculate the proportion of fragments longer than 100 and 200 nucleotides. Based on the QC metrics, identify the samples with fewer than 40% of the fragments longer than 100 nucleotides to allow exclusion of these samples from